We whipped through a couple things real quickly at the end, but I just want to kind of go back through and kind of address them. A lot of it is just rote memorization stuff, which is why I felt it was okay to kind of just gloss over. But we should at least officially mention it again. And for those of you looking for the Unit 3 slides, I apologize. I literally just posted those. So if you check now, they are there. I will also say, as a word of caution, it is a massive file as far as PowerPoints go because of slide number 2. So if you do decide to print them, you will probably want to download it first, delete that slide, and then print. If you delete that slide, it goes from probably 35 megabytes to 35 kilobytes. It's a massive image, and we'll see it pop up here. But anyway, regardless, disaccharides. Disaccharides meaning di, two, saccharides are carbohydrates. So there are some common ones that pop up. You get sucrose, lactose, and maltose. And since it's actually right here, we can look at it. Sucrose is our standard table sugar. That's the one that we process really, really, really well. Okay? And so when we link those, we're linking glucose and fructose, and they're linked via an alpha glycosidic bond. I remember the alpha is the higher energy one, right? The less stable conformation of our structures. So that sugar gets processed relatively quickly and easily. If we take a look at lactose, it's galactose and glucose. Okay, so again, we don't need to know specifically what those structures are, but at least recognize those patterns. The interesting feature here is how do we link galactose and glucose when we're looking at the disaccharide lactose? It's linked in a beta 1,4, meaning we aren't looking at the alpha conformation of those sugars anymore, we're looking at the beta conformation. And what do we say about the beta conformation? It's more stable, which makes it more difficult to process. Anybody lactose intolerant? Why would we be lactose intolerant? We don't have the enzymes to properly process the beta form. Okay? And so that can cause problems with digestion. Maltose is 2 units of glucose linked with that alpha form again. Okay? Apparently I'm not hydrated today. So we can also look at our disaccharide structures, so we can get our nice pretty pictures of these. You'll notice that we have a couple different kind of formations. Notice what we're saying at the top there with our lactose. That bond goes, goes up, then down, and then up. What is happening at that point? Everybody at least see where I'm pointing? Notice that the way it's drawn, it is a point. What is at that point? Nothing. Nothing is at that point. Okay? What are they trying to show? That oxygen is on the upside, so that we can then say that our CH2OH and that oxygen are on the same side, making it cis or beta. So what they're trying to show with that goofy bond, okay, in some cases you'll notice our textbook does a horrible job of it because they make it a point, and points by definition are carbon, okay, again from organic chemistry. So our online homework switched it a little bit, and instead of making it a point, they show it as kind of a goofy curved thing. Okay. We're trying to keep the sugar in the proper conformation, in the Hayworth projection, so it is easier to interpret the L, D, alpha, and beta. In so doing, we have to make manipulations to our structure so that we can see that linkage a little bit better. Kind of makes sense? Okay, so just be aware of that as a notational mess in biochem. So it just comes from we just accept that. Okay, so there are our sugars. I won't test you specifically on it, though I will say it is probably really important for you to memorize the structure of glucose. And the structure of not one on here, ribose. Okay? Those two structures are super important. Glucose because, well, it's the primary energy store molecule. Okay? So glucose is an important structure that if you're moving into kind of standardized tests, they will probably expect you to know the structure of glucose for that reason. Ribose, why would you need to know ribose? DNA. The carbohydrate in DNA is ribose. So knowing those two structures is going to be kind of important if you're moving into standardized testing. Okay? I think it's a bit silly to memorize stuff, so I don't specifically test on it. I'm just making sure you're aware that those are important sugars to know. Make sense? Our polysaccharides, cellulose. Where do we find cellulose? Plant material. And it is there as a structural feature. Okay? So if it is structural and we're looking at carbohydrates, what kind of linkage would we expect in those polymers? We would expect that beta linkage because the beta linkage is more stable, okay? And that means it's harder to process. It's harder to tear down, which is exactly what we want for structure. We don't want it to just fall apart. Okay? That's the biggest part of it, in my opinion, that you should know. The rest of it is, again, just a bunch of lists of facts. They're talking about how it contributes to structure. Number one, beta is super stable. And then number two, if we're looking at that carbohydrate chain, okay, our polymer chain, what do we have hanging off of both ends? And by both ends, I mean the top and the bottom of the structure, not the ends. So top and bottom. What do we see at the top and bottom of that? We see hydroxyl groups. Okay, well, what is special about hydroxyl groups?
They can form hydrogen bonds with their...
We can generate hydrogen bonding. Hydrogen bonding is what type of force? Let's look at strength in that respect. What kind of force do we get from hydrogen bonding? Strong force or weak force? Strong. It's one of the strongest forces we can possibly get. So when we're looking at our carbohydrates, they have these OHs, which does a couple things. It can open up sites of reactivity. Okay? In this case, with most of our cellulose structures, it's not enough to be reactive. But what it does allow for is strong intermolecular forces. So that if I layer a second polymer strand on top of this, I get lots of hydrogen bonding interactions between them, which then means those strands are very tightly connected to each other. Well, the tighter connection they have, the harder it's going to be to pull them apart. Okay? Kind of like using a screw instead of glue. Okay? The screw has a tighter interaction with the two materials. That holds them together better. Okay? So we get a better structural feature. So if we can increase the strength of the interactions between those polymer chains, we get more and more structural features coming out of this. Starch. What's the deal with starch? Where do we find starch? We could cheat. Plants again. But what is the linkage in starch? It's alpha. So it's still a sugar glucose chain. Okay, but instead of it being beta, it's now alpha. What does that mean for the role of starch? It's an energy source. It's not there as a structural feature. It's there to provide energy. Okay? That makes the anomeric carbon much more reactive and allows it to be processed and broken apart to get access to the individual sugar molecules or the individual glucose molecules. Okay? Where do we find starch? Yes, plants, potatoes. What do we use potatoes for? Food, energy, okay, because we're getting access to the starch molecules, and the starch molecules provide us energy. When we go through and look at those linkages, we'll come up with a couple different forms of starch. We have amylose versus amylopectin. What's the difference? Say that again. What one connection?
The amylopectin.
That one?
No,
That one?
no, the other way. Keep going.
Up there?
No. That one.
We've got another type of linkage here. Okay, what we're linking now is two chains to each other. Okay? That's going to compact the amount of energy into a smaller source, because now instead of having those free-flowing chains, they're now linked to each other. Okay? Interesting issue with linking it that way, though, is we're now taking the anomeric carbon and we're burying it into a harder to access structure. That makes that a little bit more difficult to process. So when we're looking at our linkages, we don't have just 1,4, we'll also have 1,6 linkages. Okay, well, that's going to change the structure. Well, that changes the structure. How do we get access to that molecule? Well, we'll need a different enzyme. Right? So that enzyme processing then comes in when we're looking at the decomposition, and that's where we run into amylases. So the amyl is again referring to our amylose and amylopectin. The amylase, our ACE, is that enzyme ending saying it's going to cleave it and cut it open. Okay, because we had two types of linkages, we're going to need two types of enzymes. Okay, so we can end up looking at, I'm pretty sure, yep, I'm positive that we have an alpha and a beta in here, and I do not remember which one's which. The beta's on top. Thank you. We have the beta amylase, which is our exoglycosidase. Exo being outside. So that enzyme is going to work on the outside of our structure. Okay? So that's going to start to look at our 1,4 cleavages, the ones that are more accessible. The alpha is an endoglycosidase, meaning it's going to go for the inside or the internal ones. Okay? It will start to cleave those 1,6. And it'll start to cleave anywhere along the chain. Sorry, the 1,6 is a different one. Anywhere on that chain. So we get two different types. One that just works at the ends, and one that works in the middle. All right, why do we want two different types? If we just worked at the ends, we're going to produce energy. Right? We're going to get glucose molecules. But how many ends are there? There's 2. Okay, so yes, I can produce energy, but I can really only produce sort of 2 energy equivalents at a time. What happens if I have an endoglycosidase? Well, that can go somewhere in and cleave that chain in half. Now how many ends do I have? 4. 4 ends means 4 energy units that I can now get access to. Okay? So we want the ability to say, oh, I don't need a lot of energy, just give me a little bit. Oh, I'm going to cleave off just the ends. Oh crap, I have no energy, start cutting that crap up because I need something fast. Okay? And that's where it'll start cleaving and breaking it apart into the smaller sections. Both of those are going to end up cleaving at our 1,4. Okay? So we need another enzyme to cleave the 1,6. That's going to be your debranching enzymes. Okay? Debranching because, well, what happened with our structure? We've got a chain that then branches to another chain. Okay? So we need a special enzyme to cleave that branch. Okay? Neither of these cleave the beta conformation. They'll only cleave the alpha. Okay? Why, again, would we want that? Beta is part of the structure. We don't want to break the structure. Once the structure is built, it should be relatively permanent. Animals don't have starch. Okay, so how are they going to store? Well, through something very, very similar. We call it glycogen. Okay, it's the animal equivalent of our amylopectin. Okay, we're looking at 24 to 30 units of D-glucose. Again, the polymer backbone 1,4 glycosidic bonds and branches 1,6 glycosidic bonds. Okay, which seems a bit weird because it sounds awfully familiar to amylopectin. Okay. Take it a step further, what do we need to break those down? We need the same enzyme. But it can't be the same enzyme because the enzyme used to break amylopectin is from a plant. The enzyme used to break glycogen is from an animal. So it can't possibly be the same enzyme, but it's going to be a derivative thereof. Okay? Which is then why we invent new words and new definitions or new names of these things. I believe I have it in my notes. I actually just say the equivalent of our amylase. So we end up with another enzyme that goes through and cleaves that. So they're different, but the same, but different. Okay? Chitin. Where do you find chitin? Oh, sucks. Okay, wait a second. Where do you find chitin? Okay. Chitin is our or not cell walls. This is our invertebrates, insects. That hard crunchiness when you bite into a cockroach. That's you breaking through the chitin. Sorry, I thought that was worthwhile. Believe it or not, where are we potentially going with our food supply? We're going to insects. Okay? Insects are in a much larger abundance than animals, okay, than cows, and believe it or not, are a better protein source than cows and chickens and all that fun stuff. So, what's that? And a lot of other cultures already eat them because they're much more readily available and they're easier to process. Okay? I know it sounds disgusting to us. I had termites. Termites aren't bad. It actually tastes kind of grassy. Yeah. You just stick your finger in the nest, they crawl over your hand, you just... Yeah. I said I didn't want to eat a stick. So chitin, being a structural component, is also a carbohydrate. Okay? Again, we're going to be going through beta glycosidic bonds. Okay? But if it was just a beta glycosidic bond, wouldn't it then be the same thing as cellulose? Why are we then calling it chitin? Okay? Well, the exoskeleton does a little bit more because the exoskeleton actually has, I know it's not the greatest term, but a better crunch associated with it than, say, plant material. There's a little bit more of a give. Well, why would it be crunchy? Okay, what does that mean about the linkage between those strands? It's stronger. How can I make it stronger? There's two things. We could potentially do branching, or we increase the strength of the force. Well, we already had hydrogen bonding. Okay, so we could potentially go to ionic, or we could go to more hydrogen bonding. And what's changing? We're making these now the amino acetyls. The amino group is roughly the same as our alcohol, okay? But we also add the acetyl. That acetyl oxygen allows for more hydrogen bonding. So we can get a stronger interaction between our chains. That stronger interaction leads to a more rigid structure and a crunch. Okay? Cell walls, same kind of idea. Okay? If we have a cell wall, we want something to act as a barrier. It's again going to be a carbohydrate, but they've adjusted, or cells have adapted to insert other little sections to it. Again, changing the intermolecular forces. By changing those intermolecular forces, it changes the properties. Notice you're looking at a pretty significant change. The carbohydrate, we're adding what they're labeling as NAM or NAG, which is our N-acetyl muramic, I think, acid. Okay. So if we go through and take a look up at that thing, I know it's not easy to see, but from one of our oxygens, we attach a COO minus. What is a COO minus? It's the conjugate base of an acid, so there's our acid portion. We also have the N-acetyl group. So we've increased hydrogen bonding significantly now to help kind of increase the interactions between those chains. That will help contribute to cell walls and actually make a better structure. Okay? Notice that we'll add all sorts of other things to it. We're linking amino acids out here, alanines. Okay? We're linking, I can't even read what those things say. More peptides. Okay? We can link out lots of peptide chains off of this. Well, why would we want our cell wall to have peptide chains coming off of it? Interesting question. What is the goal of a cell wall? Structure. Is that it? Protection. So if I want to protect myself, all I need to do is build a wall all the way around me. And I will now be forever protected. What's eventually going to be a problem? What's that? I don't have any water inside my wall. Eventually I will die inside my wall without any water. Okay? What else would I need? Energy, nutrients, food. Okay? So when we're looking at a cell wall, yes, it contributes structure, but it also must allow for some things to pass through. Okay? So what would facilitate potentially the transfer of different pieces? Maybe like an enzyme. Well, what were enzymes made out of? Proteins. What are proteins made out of? Amino acids. Another name, peptides. So by linking peptides to our structure, we can build markers that can say, hey, actually, this is, I've got a little handout here. Whenever I see water, grab water and bring it in. Water's going to pass through regardless, but it works. Okay? Or a nutrient. Oh, that's a useful piece of food. I want that piece. I'm going to take that. Okay? Without those markers on the end of the cell wall, there's now nothing can really pass through. It's just kind of stuck on the outside or stuck on the inside. We need something to facilitate that kind of marking and transferring process. Right? Which I believe leads us into, not quite yet, Glycosaminoglycans. Okay? Glyco, sugar, amine, our nitrogen, and then glycans, potentials for more sugar, so how we're linking those. So these are polysaccharides based on repeating disaccharides, where one of the monomers is an amino sugar. Okay? These are listed as potential molecules. You've got your heparin, hyaluronic acid, sulfate, carotene sulfate. Okay? So these things help contribute to a little bit of structure and signaling. And this is really where I thought we were going, our glycoproteins. And if we take a cell wall and we link on some sugars out at the end of our cell wall, what could those sugars potentially do? Well, they contribute to structure. They also have, dumb statement, I'm going to say it again, structure, but that structure is now different from other sugars, which means that sugar chain can now act as a marker. Okay? That could act as a marker to say anything that I make has that stamp on it. I now know that that's something I made. So, well, how is that a big deal? Okay, well, are there things out in the world that get made that could potentially be dangerous to us? Well, if we didn't make them, we want to be able to mark it and say, hey, that's bad. Get that out of here. Okay? We can do that with our blood. Our blood has varying types. We have O, A, and B. What are those in reference to? They're in reference to a carbohydrate chain hanging off of our cell or our blood cells. When we mix in different types of blood, what happens? Okay, well, our immune system says, oh, that's the O marker. I make O, that's fine. Okay? That's the A marker. I don't make A. That must be bad. Kill it. Okay? This is why when we're looking at blood, we have different types because we want to make sure we inject people, or not inject people, transfuse, is that a better word? Transfuse the appropriate blood, because our body will recognize the other markers as something bad and try and destroy it. The whole point is to still allow for oxygen transport. If we start killing off those cells, that becomes a problem. Okay? That goes back to the carbohydrate chains. So, kind of a neat feature, I think, of carbohydrates. Is that a question here?
No.
So now we're officially into the new stuff. Right? In the new stuff, what we'll be looking at is reactions. Right? Reactions across potentially all cells, and this is where we run into potentials. So one of the things we want to be able to do is to look at a reaction and label it and say, that is this type of reaction. Because we all had a class where you went through and did that at a simple level. That was organic chemistry. What organic chemistry should have given you is what is an acid-base reaction. Or really, Gen Chem should have given you that. But then we have substitution reactions. What happens when we run a substitution? What happens when we run an elimination? What happens when we do an addition? Okay? Biochem brings in a couple other ones. Okay? We get isomerization, which is kind of a reaction and kind of not, and then reduction and oxidation. We didn't typically look at reduction and oxidation reactions in OChem because we could kind of label them into the other ones, and we had to transfer electrons. And transferring electrons is usually a giant mess that organic chemists don't want to look at too much. Okay? Super important, but we kind of ignore it. We just kind of accept it. Okay? Reason why? For most reduction oxidation reactions, what did we need? Think of a reagent that did reduction or oxidation. Either one. Okay. For a reduction, I heard LiAlH4. Lithium aluminum hydride, yes. That's a reduction. Or does a reduction. Sodium borohydride will do that. Other reagents? Sodium borohydride isn't going to be as helpful. The lithium aluminum hydride helps us out. Fits the pattern. Any oxidation reagents? You remember? Really, from OChem, there's only two that you were really supposed to know for oxidation reagents. My heart's dying inside, Jen.
Yeah, I know. Sorry.
Politically correct.
Oh, PCC.
PCC, politically correct chromic acid is how I like to refer to it. Chromic acid. Other reductions, H2 and, say, palladium or platinum. What do most of these have in common? Palladium, aluminum, PCC has the C, chromic acid has the CR. Almost all of these involve metals. Okay? What is special about metals, particularly when it comes to organic chemistry? Metals aren't organic. So they typically get ignored in OChem. They come back pretty hard in biochem because in biochem we have to run reduction and oxidation reactions. So what facilitates those reduction and oxidations? Well, that's going to be metal ions. So biochem is now an interface between inorganic metals and organic chemistry facilitating those electron transfer reactions. Right? Because electrons inherently aren't stable, we need something to facilitate that transfer. That's where we get metal clusters and all sorts of other fun things like that. Okay? So being able to look at a reaction and say, that's what happened, is an important thing to go through and do. Okay? That said, that's probably because of my organic chemistry background. Okay? Do you guys recognize common patterns within these that you could say, okay, that is this, without having to look at all those reactions? Because I know you guys are writing all those reactions down. What happens in acid-base? We just transfer hydrogen. So we don't need to draw out a reaction. If we look at the reactants and the products and all we see is a hydrogen change locations, well, that was an acid-base reaction. How about a substitution? A transfer of anything other than hydrogen. Right? That's now a substitution. We're taking something other than hydrogen, which could be an atom, it could be a whole structure, now moving locations. What's happening in the elimination? Okay? We're really making a pi bond. It is one pi bond. For most cases in biochem, and for that matter ochem, that's the formation of a double bond. Okay? But it doesn't have to be a double bond or an alkene. It could be a carbonyl, and it could theoretically be an alkyne. Okay? But in most cases, we're looking at the formation of an alkene. Addition. What's happening in an addition? We lose a pi bond. So recognizing what's written in the words is probably more relevant than looking at the pictures in this case. What we're just trying to do is say what changed. Identify that change, mark it, label it. That's it. Okay? Which gets us to the next fun slide. Oh, that's obvious. You can totally tell what that is, right? I guess it's coming this way. Is that triggering any nightmares? We have the Krebs cycle. We've got glycolysis coming into it, both of which were theoretically discussed in a biology class. So what we're now going to be kind of stumbling into is a review of that old material that you saw in biology. We'll be looking at it much more carefully than, say, a biology class. That said, when it gets tested on, it's getting tested at pretty much the level of a biology class. Okay? Do you know what the individual steps are? What is the rest of this? It's the rest of the chemical reactions that we are currently aware of as of at least 5 years ago. That's when I found this. In cell metabolism. Okay? You might say, well, how could you possibly read that? That's right, you can't really possibly read it. So we're going to wait a couple minutes here because I've got a link. And we'll go out to that link. And it's going to take a little bit to load. Escape. Go away. So if we go out to the link, this, folks, by the way, is a pretty fast internet connection. Yes, there's no way I could read that. That's why we have the Zoom feature. I completely agree with you, Mark. This is fascinating. This is absolutely amazing to look at the detail happening within this. You're getting the names of the enzymes. You're getting the reagents used to make these things. Okay? You're getting the structure of these things. Okay? The names of the compounds. Okay? This is absolutely amazing. Okay? In this one slide, you're probably seeing thousands of PhDs. Okay? And I don't mean people studying them were thousands of PhDs. I mean literally thousands of people got their PhD by looking at probably just this one reaction. Okay? So we're looking at a massive combination. So when people talk about the complexity of biochemistry, I think a lot of it kind of disappears because when you look at it, oh, glycolysis, it's this chain. We take sugar and we do this. Well, what else is happening? Let's see, did I actually find it right? I did. Wow, look at that. Glucose. What happens to glucose? Well, in glycolysis, the very first step is right here. Using phosphoglucomutase, that's awesome because we use different names for the enzymes, we make glucose with a phosphate on it. That's step one of glycolysis. And yet, where did the glucose come from? Well, it came from this reaction. It could go either way on this reaction. It could go either way on this reaction. It could go out over to here. It could go out over here. It could come from there. It could come from over there. When we're looking at biochemistry, you can say, well, I'm going to make a drug stop this one reaction from happening. That one reaction is connected out to probably 20 others. Okay? The fun part of that is we're probably only aware of 3 of those 20 others. So that when we build a drug that targets something or removes an enzyme, there's so many other connected properties within that we don't necessarily know what's going to happen. Okay? That's why we have tons and tons of clinical trials and why it takes a long time for drugs to get to market, because we have to test this, okay? Which is fun. How do we test this? We could give it to people. I've got this drug that in a cell cures cancer, yet the first person I give it to, they die. Probably not a good study, and you will probably get sent to jail. So how do we test it? Okay? We run into an ethical dilemma of putting it against people because they die and people tend to complain because people have families. Okay? So we usually end up using mice, okay? Which then, yes, is an ethical dilemma. Okay? We get PETA yelling at us. Okay? There really is no good way around it. If you're going to generate drugs that can cure things or at least help problems, there's going to be deaths involved. The question is, where are those acceptable deaths? And that sucks. Personally, I think that sucks because I don't want to be responsible for that. Okay? So just wanted to at least point that out. I think it's really neat. When we think about biochemistry, this is what a biochemistry class is supposed to teach you. All of this. There's no way in hell that's going to happen. We're not even going to get close. We'll be lucky to get through glycolysis and the Krebs cycle. Okay? But that's what we're trying to do, is get you the basic pieces that you could then apply that basic knowledge to anything in this massive web of disaster. Kind of makes sense? Is that now like, oh crap, you should have told me that day one and I would have left? Kinda. So, the part that's neat, but at the same time, the part that really sucks. So, current slide. Probably should not have started it with the current slide. Hey, that worked. So yeah, if you want to go out and take a look at that, I think it is neat. There is a link at the top of that page. This is the slide that is a pain and will take forever to download. So as soon as you get it, if you click it, you'll probably have to wait a couple minutes for it to load, but then you can delete that slide, or at least the image in it, and that should speed up your processing time of the PowerPoints if you wanted to look at the PowerPoints. Make sense? Okay, so, what chapter is this? I think this is chapter 15. I honestly don't remember now. Remember at the beginning of the semester we talked a little bit about free energy, energy diagrams, all that fun stuff. So we're going to come back and talk about that again to a little bit of an extent. We're looking at our chemical reactions. We can look at a Gibbs free energy. Okay, so a change in Gibbs free energy for a reaction, and take that back to the concentrations of the reactants involved. Okay? So what this is actually showing here is that the Gibbs free energy for any given reaction at any given temperature will be equal to the Gibbs free energy for that exact same reaction. Well, duh. Oh, but at a very special case. What does that little zero mean? Standard conditions. Right? So the Gibbs free energy at any given point will be equal to the Gibbs free energy at one point plus some variation of that. So we need to know what the standard point is. So what are our standard conditions? Certain temperature and pressure. Are we going to need to be concerned about pressure? Probably not, because pressure is gases, and almost everything we're working with is in a liquid state. It's dissolved in solution. We run into some exceptions there. Do we have pressures of oxygen? Yeah. Okay, we get a pretty large pressure issue, particularly when we're looking at lung function, because we're looking at the transfer of CO2 out and O2 in. Okay, so pressure does kind of show up. What are standard conditions beyond that? Well, pH 7 probably makes sense, right around neutral, physiological pH. What temperature do you think we look at?
37 Celsius.
37 Celsius seems to make sense, because 37 Celsius is human body temperature. Ooh. Are we always studying humans? No. So that's a bit questionable to pick 37. Okay. Not a bad idea, but we aren't always studying humans. Okay. So what might be a more consistent temperature in all of our studies? Room temperature. That makes good logical sense. What is standard temperature? It is 0 degrees Celsius. Why? Because some jackhole thought it would be funny. I don't know. That's what they picked, was 0 degrees Celsius. Okay? It is a standard, though. Okay? So if I want to say the standard height is 6 foot, guess what the standard height is? 6 foot. If I want to say the standard height is 5 foot, what is the standard height? 5 foot. Okay? So standard does not mean consistent. Standard means accepted. Well, what is accepted? That depends on the reactions you're looking at, the courses you're studying, and all of those conditions. General science runs standard conditions at 0 degrees Celsius, 1 ATM, okay, all that fun stuff. Okay. But just because that is the general standard condition does not mean it is always that. I've run into issues with this working with my wife. They were trying to figure out what was going on with their equipment because they couldn't match up temperatures and systems, or they were running calculations. So, well, it shouldn't be producing this much. It doesn't make any sense what's going on. We eventually tracked it back. In the calculations the engineers did to design the equipment, they calculated standard temperature to be room temperature. Motherfucker. That is not standard temperature. And as soon as we made the change, everything lined up. Okay? So standard condition is something that should not be changed. It is a single term, and it should never change. Be very careful if someone makes reference to standard conditions to make sure you know what they're actually specifying as standard conditions, because it's not standard. Okay? So that's our standard conditions plus some variation off of that. Well, one of the things that would vary would be temperature. So that next term has to include some kind of temperature. Okay. What else is it going to include? Well, when we're looking at standard conditions, we said 1 atmosphere. Well, that's a pressure. What are the units on things in brackets? Concentration. A molarity, not a pressure. So I said, well, how do we incorporate pressure? Well, what is pressure? Right, this goes all the way back to Gen Chem. Pressure we write as P. What is pressure? PIV by definition is not P, so right idea, solve that around. Which you should have seen at very least in 130. That may have been so long ago you don't remember. That's okay. Okay. What did N stand for? What did V stand for?
Volume.
Except volume, what unit?
Liters.
What is moles over liters? Molarity. Pressure and molarity are directly related to each other. So just because we don't see pressure obviously written in here, it is technically in there in concentration. So what is your standard concentration then? 1 molarity. Okay? That's your standard. These would be any variation thereof, or off of that. Okay? What is C, what is D? They are products. What is A, what is B? We have the products divided by the reactants. Where have we seen that before? That was the equilibrium constant. So what this equation is telling us is that we can relate the equilibrium constant, the amount products are favored or reactants are favored, we can dump that constant into this expression. When we change the temperatures, that allows us to vary off of this. Okay, kind of neat. It's actually officially Q in this case, because we aren't at equilibrium. No, it's K. Let's just say that. Let's just make sure we don't worry about that. Okay. What are the C, D, A, and B? Those are the coefficients from our balanced equation. Okay? That's why everything lines up there. So, in our thermodynamics, okay, in this chapter, what we're concerned about is the energy balances of all of our reactions. And how does that energy balance dictate what happens, okay, or if it happens. So if we look at the thermodynamics of our whole process, we start with hydrogen in the sun. What does hydrogen in the sun do? Okay. What happens? How do we form hydrogen in the sun? Okay. Or take it a step further. Is the only element in the sun hydrogen? No. Okay. What other elements are there? Helium. How did we get helium? What is helium?
You get enough energy and 2 hydrogens, not the energy.
Helium is 2 protons. How do I get helium? Well, I take 2 hydrogens and I smash the 2 hydrogens at each other hard enough that I fuse them. And I get nuclear fusion. What is special about nuclear fusion? It releases a shit ton of energy. Well, where does that energy go? Outwards. Outwards where? Passes flying through space until eventually it hits us. Ta-da! It hits us, and in particular it's hitting plants. Plants take that energy, okay, combined with water and CO2, carbon dioxide. Plants need carbon dioxide. Why? Because they take the carbon dioxide that is this incredibly stable molecule and take a crap ton of energy from the sun, shovel it into the CO2. Now the CO2 goes, oh crap, I've got too much energy. What can I do? Well, I see another molecule. I'm going to go react with that. There's step 1. We're now changing the structure of CO2. That gets metabolized into glucose. Now we have glucose. What could we do with that glucose? Eat it. We've now taken this massively stupid high energy, melt your face, bones, permanent existence, turned it into, or combined that with CO2 to make another high energy molecule. We take that high energy molecule, we eat it, and convert that high energy molecule into ATP, which is now our energy currency that we can spit out. In the process of doing that, what did we do to that glucose? Broke it down into CO2. The CO2 then feeds back into this cycle. Okay? That ATP then goes out, can generate amino acids, sugars, combine those, make a larger structures, proteins, DNA, RNA, carbohydrates. It's all coming from starting way up here. Okay? This whole process starts because we have a stupid high amount of energy. And that's the only way this works. Because we start here, and really what we should be showing here would be this process. We start at, that should be on the far left, but I don't have any space over there, a really high energy state, which then falls through space. As it falls through space, it loses energy. Then plants get it. Ta-da! Plants convert that into something else, which will by definition be lower in energy. We then take that, convert it to do our stuff, and then that, convert it to do our stuff. And at the end of that whole process, we end with CO2. That CO2, when combined back with that stupid high amount of energy, can put me right back up at the top, and I can start the whole cascade again. Okay? So when we're running these processes, each of these is resulting in a drop in energy, okay? Which is a bit of an odd concept, because that would say all chemical reactions are exothermic. Is that true? No. Well, how do we have endothermic reactions? Okay. Officially, all chemical reactions that proceed are exothermic. Why do we have endo? We're adding in another molecule of a very high energy, and we're ignoring that when we're looking at our balance of our reactions. Okay. We are only looking at one species. So when we're looking at our reactions, all of the reactions we're doing in biochem are exothermic. Okay. The only way we can get them, or the reaction to proceed, even if we say, well, but that structure shouldn't, I should never do an elimination reaction. Why should I never do an elimination? What does an elimination reaction produce? A pi bond. Double bond. That's a pi bond. Pi bonds are higher in energy than sigma bonds. Okay? So if I start at sigma and go to pi, I've moved up in energy. I can't do that. How is that reaction happening? I'm adding something in there to either drop the energy of the activation barrier. That doesn't change the energy of my reactions and products. I'm adding another species that's high enough energy that forces that reaction to occur, or I prevent the reverse reaction. Okay? All of our reactions, even when they're endothermic, we've facilitated by somehow introducing another species that allows for a shift in balance to make that an exothermic reaction. But depending on what we're looking at, we may not evaluate that aspect of it. In biochem, we're always saying, well, crap, that reaction shouldn't happen. How do I get it to happen? Well, if I don't have enough energy, what do I need to supply to get that reaction to happen? Energy. Where do I get that energy from? The answer is on the board. ATP. So if we look at a reaction and say, it's an endothermic reaction, that'll never happen. Guess what cell life has done? It has an enzyme, and that enzyme brings in ATP, forces the reaction to occur. Allows it to be overall thermodynamically favorable. So anytime you see an endothermic reaction, right, that's a big red flag that ATP is going to be involved. Okay. Is ATP the only molecule with energy? No. So we will have other molecules that can contribute. It just depends what situation we're looking at. ATP happens to be the most common. We talked about catabolism, and I can never say that word. Catabolism is breaking molecules down. The A1 is making bigger ones, so we're building up. Okay? In the process of building larger molecules, that is by definition going to be endothermic. We're making something. To make something, we have to put energy in. So that has to be an endothermic reaction. Well, if that's endothermic, well, that's not going to be a good thing to do, or an easy thing to do. What do we have to do? We have to supply ATP. So in almost all of our A-word reactions, we're adding ATP. There we go. A for ATP. We need ATP. Hey, look at that. My bad speaking works. Okay? And catabolism, what we're end up doing is breaking molecules down. Well, as we break molecules down, what's going to happen? We're going to release energy. Well, what's that energy that we're releasing? Typically ATP. Okay? Some other things that we can look at. Transferring electrons. Okay? When we're looking at catabolism, the overall process is oxidation. Okay? When we looked at that previous slide, we said we staircase down to eventually what? CO2. What happened to the carbon? It is fully oxidized. All of the bond sites on carbon have oxygen. So when we're looking at catabolism, when we're breaking something down, the end result will always be CO2. Well, CO2 is as oxidized as we can get. Catabolism is an oxidation process. Well, if that's oxidation, a word has to be reduction. Okay? So we have to remember our tying catabolism to oxidation, a word to reduction. But when we look at reaction and go, oh yeah, that's totally reduction. Or, oh, that's totally oxidation. What are we looking for to say that is oxidation or that is reduction? I think I can do it with this one. Yep, this will work. What type of reaction happened there? Oxidation. How do you know that's oxidation?
Added bond size.
Shh, I knew you were going to freaking say that. Fine. Deal with that. No bonds to oxygen. Can't give me that answer.
Removing hydrogen bonds.
Nah, I was a jerk.
Is that what you were looking for?
No. So what you guys have done is taken enough chemistry that you come up with shortcuts that you don't have to do this the official way. What is the official way? You all probably blocked it out, because I know all my 130 students freaking hate it.
Oh, you all the math stuff that you're looking for?
Mass stuff looking at what? Begins with O,
Oxidation state.
the oxidation state. What is the oxidation state for carbon in CH4? Hydrogen's always a +1, so carbon's a -4. What is it in CO2? It's a +4. Oxygen's always a -2. Okay. What happened? How do we go from a -4 to a +4? What happened to it? It lost electrons. Loss is a negative. Electrons are what charge? Negative. If you lose a negative, you become positive. So when we're looking at our reactions, we can try and cheat and use our OChem knowledge, and we'll come back to that definition. Officially, what we're looking at is the transfer of electrons. So we'd have to be able to have some knowledge of our oxidation states, or looking at curved arrows to see where electrons shift from. Okay? So we do need to remember that your oxidation process is a loss of electrons. Our reduction process is a gain of electrons, also known from the very beginning of the semester as Leo says Gur, to help remember that tie. Right? We clicking on that review a little bit? Not clicking on that review a little bit? Which part? Just remembering loss of electrons is oxidation? Okay, so there, that's literally all you need to know. Loss, electrons, oxidation. So how do you spell Leo? L-E-O. L is loss. E. That's the big thing you need to know. Tie that. Determining the oxidation states, not so much. That was the first exam, if we even did it on the first exam. I'm not even sure we did oxidation states on the first exam. Okay. But recognizing that loss of electrons is oxidation is going to be important. And if it's oxidation, we then know, fair enough, we lost electrons, and what else would that tie to? Oxidation is a catabolic process. So it's tying all of those concepts with each other. Does that make sense? Energy diagrams, yeah. Catabolism. and anabolism. So we'll always have a reactant. So since we're comparing these, let's make sure the reactant has the same energy. Okay. If it's a catabolic process, where's the product?
Lower.
It needs to be lower in energy. So if we looked at a catabolic process, that's horrible, but good enough. That would be our energy diagram for a catabolic process. How about the anabolic process? The product is higher in energy. We now have an anabolic process. Okay. To get the anabolic process to work, we'll end up having to add another reaction to this. We'll have to add ATP going to ADP, because that reaction is ATP to ADP? Exothermic. So if we add that reaction in there, let's just say the reactant's the same energy, our product energy is somewhere down here. What does that mean? For the overall reaction, the red one is a positive value, the purple one is massively negative. When I run those two reactions simultaneously, the overall result is one that is negative. Now that reaction proceeds. Does that make sense? So we're looking at an additive process of running our reactions. The anabolic reactions do not happen well on their own. Okay? We'll have to run them simultaneously as a very exothermic reaction. Okay? So that the net balance is one that is exothermic. That does not make it catabolic. Okay? Kind of make sense? Okay. Do I have a GIF on this one? I did. This is how I remember metabolism. Right?
Oh, Anna makes her cat breaks.
What's that? What was yours?
Oh, well, my micro teacher taught us Anna makes her cat breaks. So, like, cat breaks down.
That works.
I don't know.
Which is
That's
kind
what
of
you were
similar
referring
idea.
to with the
Yeah.
cat.
Anna makes cat breaks. Right? See the cat breaking stuff. Breaks. It just happened to stick in my head. So, what the hell? Let's just ignore that. Our oxidation states, this was the slide we had at the beginning, wasn't it? Because I totally thought this was a slide we already talked about.
No.
That would be the explanation for why we missed that. So I apologize. I totally thought this slide was already at the beginning of the semester. So when we're looking at our catabolism and anabolism, for those of you who missed it, so we step back. How far back do I gotta go? We'd have to look at an individual reaction and we'd have to decide the oxidation states. So I will have to explain this one now, probably again on Thursday. We have to determine the oxidation states for each element on both sides of the equation. Okay. Because it's biochemistry, we really only have to simplify it down to one big rule. The oxidation state for oxygen is -2. The oxidation state for every other element must be calculated. Well, how do we calculate the oxidation state? The oxidation state is the balance of protons and electrons on an atom. What is the charge? What is the charge on this first molecule? Your charge is always given to you in the upper right-hand corner. So what is the charge on that molecule? 0. But how do we determine the charge? What is charge? If an atom was negative charge, what does that mean? It has extra electrons. If it's positive charge, what does that mean? It's missing electrons. Why does that make it negative or positive? Wouldn't it, if it had electrons, it'd be negative? If it didn't have electrons, it would be positive?
The overall balance between protons and electrons.
The balance of protons and electrons. But wait a second, what did I say oxidation state was? The balance of protons and electrons. So I'm using a very similar definition for both charge and oxidation state. The difference is that charge is the balance of protons and electrons on anything. Oxidation state is the balance of protons and electrons on an atom. Okay? So if we take a look at that first structure, go away. What is the balance of protons and electrons in that structure if we're looking at the charge?
0.
The charge is 0, which means I have the same number of protons and electrons in the compound. Okay? But each of those elements contributes an oxidation state, which is its balance of protons and electrons. So when we're looking at our charge, it's the sum, that says sum. Sorry, let's try that again. That fun mathematical symbol. Sum of the oxidation states will equal the charge. So we know in this case the charge for our molecule is 0 because it's in the upper right-hand corner. What is the charge on oxygen? That's what I told you had to memorize. But how many oxygens are in that formula? 5. Every single one of those oxygens contributes a -2. So I could go through and add -2 a whole bunch of times, or I could just say 5 times. I then need to add to this the oxidation state for the iodine. Well, what I said was you have to calculate the oxidation state for iodine. Don't have a choice. You have to do it. So it is unknown. It is X. We have to solve for it. But how many iodines are there?
2.
Can I solve for X? Yes. When I now go through and set this up, what do I get?
5.
Should see that X equals +5. That's the oxidation state for the iodine. Okay? The oxidation state for the oxygen, as long as it's in a compound, is -2. Okay? It has to be in a compound. Carbon, +2 to +4. What happened to the oxidation state on iodine? It went from a +5 to a 0. How do we go from a +5 to a 0? What changed? It had to gain electrons. So that's where we come up with the, did I have that there? Just erased it. Gain of electrons on the right. What happened to the oxygen? There was no change. The carbon lost electrons. Now that we know what happened to the balance of electrons for each of those compounds, we can then go through and label them as oxidation and reduction. That's where we end up with the loss of electrons oxidation, gain of electrons reduction. That's the Leo says Ger. I appear to be one of a few people that does Leo says Ger. The other one that pops up, you might have heard oil rig. It's the same idea. Okay? This is a biochemistry class all about life. We don't want to look at death with oil. So oil rig is stupid. You're dumb for using it. No, you're not. Whichever one works is fine with me. But you need to tie oxidation to loss of electrons, tie reduction to gain of electrons. Okay, I will rework that on Thursday.